Good morning, class 11, my children. Today we will go through the secondary growth only found in the dicot stem. Children, as the name suggests, secondary growth, that means not primary it is. That means not essential growth of a plant. It is a secondary growth, yet it is essential for the big trees, but it takes place after the primary growth of full body. Then it leads to the secondary growth, and secondary growth always takes place in the dicot stem, not in their root, not in their leaves, and not in the other parts of the plant. So secondary growth found in dicot stem, and today's topic is the same. Okay children, now secondary growth. So when they have or they develop their secondary growth, so they have to produce some new cell to widen the girth or the girth of the stem, and due to this secondary growth they have to manage some amount of cell which can grow or which can develop into the widening of their stem or diameter of the stem. And after that, if it occurs, so not if, when it already occurred in the dicot big trees, then they should have some bark over it, so it can't be damaged from the outer source of any type of harm. So that's why they have the secondary growth. Secondary growth, as per our study, it is divided into two halves. We can study by the two in a different way. First is stellar region, and second one is extrastellar region. Stele is the center part of the stem, which consists pith and vascular bundle, xylem and phloem. So this is the stellar region, and extrastellar region, also called as phalogen, it consists cortex, then or you can say the cortical cells, and after that epidermis. Children, epidermis, when the plant gets mature, then it is called as periderm, and that other areas rather than the stele. So this is known as the extrastellar region. So today children, we will start about the stellar region. So first step in that stellar region is origin and formation of vascular cambium. Cambium tissues you studied in the lower classes, 8th, 9th or suppose 7th. So cambium tissues are either square, not every time square, but it is rectangular. Plate-like cells are present which grow in between the, first it starts to grow in between the xylem and phloem. So that is the origin and formation of vascular cambium. So these cambium cells, when they start to grow between the xylem and phloem, at that time our in that vascular bundle, we can say it is the open type of vascular bundle. And where there is no growth of cambium cell, it remains only xylem and phloem, so they are known as closed vascular system, not system, bundle. So mainly monocots have closed vascular bundle and dicots have, because dicot have secondary growth, they have to produce the new cambium cell, so they have open vascular bundle. So first, the formation of vascular cambium, interfascicular cambium and intravascular cambium. First, the cambium cell will grow in between the xylem and phloem, so it will be the intravascular cambium. So in all the vascular bundles, when it grows completely, then in between the two vascular bundles, there will be the growth of infravascular cambium. Then it can form a total circle in a stem, and in this way they can complete the thickness in total stem, and in this way they can increase the thickness of the stem. So first they will produce interfascicular cambium, and then they will produce intravascular cambium, and it can form a ring. Next one, structure of vascular cambium. Now how the vascular cambium grows, how it starts to grow. So they grow in the two ways. First is fusiform initial and second is ray initial. As children, fusiform you know about the taproot system. Fusiform have the tapering end at one place, or at the end there is a tapering should be. And another one is ray initial. So first we will go for the fusiform initial. They will help to form that secondary xylem, that means xylem trachete mainly, and phloem cells. So these are produced by the fusiform initial. Mainly they are xylem trachetes and with the tapering end, this fusiform initials, and ray initials, they will produce the extra, or elsewhere there are parenchymatous, either xylem parenchymatous cell or phloem parenchymatous cell. Both the cells will be formed by the ray initial. So these two are the types of formation of the cell, that is fusiform initial and ray initials. And with this the secondary cells will form. Now children, third step, activity of vascular cambium. This activity means now they become active to produce more number of cells, and this earlier if it was only the one row of the cell, now they will make it more, and more layers of the cambium cells are present there, and it is done by and it is called as secondary medullary ray. When they form more number of cambium cell, and then it is known as secondary medullary. Now fourth step, secondary xylem. When the secondary xylem starts to grow, then they are axial or radial system. That means they grow either in the axial system, that means perpendicular to their point or attachment, or they start to grow in the radial system. That means radially they start to grow number of xylem cells. Now in the next step, annular rings or annual rings. So when in this way the total xylem will grow along with the full circle, then they will form the ring, and this is known as the annual rings. This annual rings, if it forms in the spring season, so in the spring seasons cells are lighter in color because there are not too much material in their cell sap, and new cell will arise. They have some transparency along with the minerals. So it is known as the spring wood, and they are light in color. And they are very far away from each other also. Next to it, these annual rings, when start to form in the winter season, they are known as the late annual ring, or also from the autumn wood, or it is dark in color. So when this new xylem will appear in the winter season, yet they will also form the annual ring. But this annual ring will be darker than the previous one because they are more compact according to location, and they are having more mineral in their cell sap to impart the dark color to them. So that's why the annual rings in the two phases, one is in the spring season, another one is winter season. Winter season, the annual ring will be darker than the spring season. So in 1 year we can count 2 rings, annual rings in any wood. So by counting, if there are 12 rings are there, so either you can count 12 then divide by 2, that means 6 years old is the, or 6 years old is the age of that tree. Or suppose you can count any dark one, so you can count the dark rings, there are 6, so it means the same result, that tree have 6 years age, or it is 6 years old. And this is the name of the branch. In that branch we can study about the age of the trees, or you can see age of the wood, yet it is still living or it is dead. By getting this ring we can consider about the wood, that how much old it is. So this is known as dendrochronology. Now heartwood and sapwood. Heartwood and sapwood. Yes children, heartwood is also known as duramen, and sapwood is also known as albumen. Heartwood and sapwood it is formed earlier, so this is having the light colored wood. Wood is already formed. Here I am not now telling about the counting the age. When the wood will form, when it is light in color due to the presence of some material, it is known as duramen, and sapwood having the albumen. This is the name of that sapwood. And sapwood mostly they have living cells, and heartwood they have some dead cells, composed of dead cell. And this heartwood, this one, if it is dead cell, having dead cell, or living cell going to be dead, at that time there will be the deposition of suberin. Now children, when there will be the deposition of suberin, so suberin makes them impermeable to water, and when it becomes impermeable to water, then slowly this wood changes to the bark. So this is the heartwood. Now according to 7th step, porous and non-porous wood. If the wood is having vessel. While it was forming the wood, at that time xylem vessel was living, and suddenly it becomes over, or suppose it was not died. So at that time, as we are cutting some section of a plant and plant root, stem, leaf, whatever. So at that time, if the formation would takes place and vessels were there, so according to that area of the vessel, it becomes porous wood. So we can see some holes on it. So this will be the porous wood, and non-porous wood, it is ring and diffuse porous wood. Non-porous wood is of two types. As the name suggests, children, non-porous, it means there are no pore. So this wood is not light in weight, heavy in weight, because there is no pore, and this is known as ring non-porous wood or diffuse non-porous wood. Children, if the name is given ring porous wood, so children, you can understand about the if it forms the ring, then you can understand about the ring, that means fully completed non-porous area, but it is diffused. And if it is ring, then it is completed, and if it is diffused, then sometimes the vessels were alive when it already made as wood, and then they become finally changes into wood. So then it becomes diffused non-porous wood. So children, for that secondary growth, now today I will taught you about the stellar region. So first it will take the origination and formation of cambium, either inter and then intra. Then structure of vascular cambium, it will be formed by the fusiform initial or ray initial. Then total formation will be cells will be there. Then we will discussed already about the activity of the vascular cambium. Activity of the vascular cambium, all the vascular cells will be having cambium rings, and these cambium rings always formed by the secondary medullary ray. Then fourth one, secondary xylem. After getting secondary medullary ray, the type of xylem, secondary xylem will form, either it is on the axis, that means axial system, or maybe it is the radial system. Then by the formation of total secondary rings, secondary xylems, they will form the annual rings. Annual rings, it is made up of two rings in one ear. Spring wood, it is lighter in color and always it forms in the spring season. So the cells are wider, broader in structure, they have less content, so that it will give the light colored area. And late or autumn or winter wood, it is having dark color due to the presence of more element in the cell, and they are very compact. They are not free from each other. So they will form the annual ring, which is dark in color. So by the counting of these two rings, we can find the age of the wood. And this is the dendrochronology. When we will study about the age of the wood, type of the wood, and variety of the wood, all these things are studied in the dendrochronology. After then, next step, heartwood and sapwood. Heartwood is also known as duramen, and sapwood also known as albumen. Sapwood is having some living cell, and heartwood is having dead cell. So this is quite very hard, and this is very soft wood. And seventh one, porous and non-porous wood. porous wood, if it is there, that means while the formation of wood, they had xylem vessel, and when these xylem vessels dried up and then they form the pores, that's why these are known as porous wood. And non-porous wood, that means there will be no vessels while formation of the wood. So they will form the non-porous wood. Non-porous wood will be of two types, ring non-porous wood and diffuse non-porous wood. If it is ring, that means it will form the full ring of that secondary growth, that means cambium, and there will be no pore, that means there will be no vessel. And diffuse means sometimes it diffuses with some other cells and then they will form the diffuse non-porous wood. Okay children, for today it is over. Thank you. God bless you.